Don't leave.
Wonderful. So are we? The meeting is okay. The lady doesn't say anymore. The meeting is recorded.
Everything you are saying now is being recorded for posterity.
All right. Thank you so much, Matt. So first of all, thank you everyone for coming. Thank you for your patience to dealing with our technical difficulties. Now we have our beautiful recording of our fantastic speaker today. So it is my great pleasure to welcome Mike Murrell from Yale University here today. Mike is a very distinguished cell biophysicist that I'm very happy to bring to you here today to speak about his latest research. He has done his undergraduate studies at Johns Hopkins in bioengineering and his PhD at MIT, also in bioengineering. After that, he has completed a joint postdoc with Margaret Gardell at UU Chicago that many of us in CSMR know very well in her research. And also with Cécile Sykes at Institut Curie in Paris, in France. He is, then after that, he started his lab at Yale, where by now he's associate professor. And his research focuses on understanding or revealing mechanical principles of major cellular processes such as cell migrations and cell division. He studies these processes on both cellular and tissue level. So I think today we will hear more about the tissue level approaches. And specifically, most recently, he has focused on understanding the non-equilibrium physics underlying biological self-assembly processes in systems. And that's something that's near and dear to us in CSMR, as many of us are in some way or the other super excited and curious about such processes. So without further ado, Mike Murrell.
So thank you for having me. It's of course an extraordinary pleasure to be here to present to this audience. So as was mentioned, the focus of my laboratory is on trying to understand fundamental principles that underlie a variety of essential biological behaviors. So we're very interested in understanding what drives cell division, cell motion of various kinds, shape change. Essentially, what we're most interested in understanding is what makes these processes very favorable, what makes life favorable, and what makes it robust. And so we take the perspective then that biological systems can be thought of as being far from thermodynamic equilibrium. We know that they can consume energy, they can perform work, they dissipate energy. And so a few of our goals then are to try and estimate something like a distance from equilibrium for different biological processes and correlate it to biological outcomes. We pursue studies into the mechanical machinery of the cell, and so we actually like to correlate these non-equilibrium aspects then to biological assembly and the performance or actuation of these machines. So first, I would like to convince you, if you already weren't convinced, that cells are amazing mechanical machines. So we know that they will convert chemical energy inside of their bodies into mechanical work. The forces that result from them can be distributed over a wide variety of time scales and length scales. And we know at the cellular level, cells can migrate through their extracellular environment by reaching and pulling and grabbing, changing their shape. Collectively as tissue, we know that the cells can move as a collective. Cells can also organize their intracellular components in complex patterns, spatial and temporal patterns, like these kind of psychedelic images that you see here that precede cell division. It's thought that these frequencies are very important for a cell to divide into. And so clearly all of these different behaviors are driven very far from thermodynamic equilibrium. But what drives them? So we know that there are energy-dense chemical compounds in the cell. So an example of that is ATP. ATP, for your introduction to biology, people call this the energy molecule. There are, of course, others. But energy is stored, for example, in these phosphate bonds. And so through hydrolysis, then, this energy can be released and can be available for a wide variety of the cell's different processes. To put it in context, the amount of energy that can be released from the hydrolysis of a single molecule of ATP can be on the order of about 20 kBt or so. Now that energy then can go on to power a wide variety of different processes, what we call active processes. So it can be used to power ion channels, which can then create gradients in ion channels on either side of the membrane. That, of course, is going to contribute to things like the establishment of osmotic pressures, changes in volume, large mechanical changes at the cellular level. We know that ATP hydrolysis is what's going to be used for the assembly and disassembly of protein-based structures. So the principal mechanical scaffolding for the cell is a network of heterogeneous protein polymers called the cell cytoskeleton. So these are each monomers, they're going to polymerize together, and that's going to be what gives the cell its shape and coordinates a lot of its mechanical activity. And so the hydrolysis of ATP is involved in the assembly and disassembly. And of course, there are other molecules inside of the cell, like mechanochemical enzymes, also known as motor proteins, which will turn that chemical energy into mechanical work by binding to filaments like this and then sliding them relative to each other. And so all of these different active processes together kind of cooperating are what drives those various shape changes, motions, and things that we see on larger length scales. To also put it into perspective as to how much energy is going into these processes, we can actually estimate from the migration of a single cell. So here is an image of a fish keratocyte. These things migrate by assembling that protein-based cytoskeleton up against the cell membrane. And so it's going to move at some speed, and the way it moves, it's going to assemble that each of these protein polymers up against the membrane. Now, to be able to move at the speed at which it does, it's going to have to assemble these polymers at that rate. Now, each step of assembly, the addition of a monomer to a polymer, is going to involve the hydrolysis of one molecule of ATP. There are many monomers in every polymer. And as you can see, there are many, many polymers inside of the cell. And so what that means then is that the cell actually has to consume about 10 to the 5th molecules of ATP per second just for that process. And so that sounds like a huge amount of energy, but of course the cell is doing other things besides just assembling its machinery for migration. I've heard that there's things called metabolism and other things that the cell also does, which are mechanical in nature per se. And so that only constitutes about 1%. And so what that means then is if we know how much, and each is about 20 kBt of energy, if we were to think of that as a thermodynamic system, then that is about 10 to the 7 kBt of energy per second, which sounds like an extraordinarily large amount of energy. We can actually think of that then as the amount of energy then that it takes to drive the system far from thermodynamic equilibrium. If you weren't doing that, if you weren't consuming that ATP, that would lead to death. And so in that sense, equilibrium means death. So we could think then as this investment as something like a distance from equilibrium, what would make it be alive, or if we were to take these components outside of the cell, what we would call, say, active. We could think of that, for example, then as not only the distance from equilibrium, but each of the subcomponents, like the amount of energy it takes to assemble the machinery, is then like an energetic cost. As that's what drives the system away from equilibrium, we could also think of that as the energy dissipated. However, this is not a system that is decaying towards equilibrium. It's one that's maintained away from equilibrium, and so we call this a driven dissipated system. So what do we know about the flow of energy in non-equilibrium systems? So of course, out of equilibrium, many of our assumptions have to be reevaluated. We know outside of equilibrium, of course, ergodicity is broken. The second law needs to be reevaluated with an open system in which energy can flow, which means that a lot of our variables are ill-defined. Things like temperature, things like pressure, which then means that there should be no equations of state that allow us to relate these kind of bulk parameters, things that we might be interested in to understand how cells and tissues assemble or move. There has been a lot of work, of course. This is a very incomplete list, but has been a lot of work to try and understand, for example, what governs dissipation far from equilibrium. And there's been suggestions alternately that, for example, dissipation can be optimized, can be maximized or minimized. One such example, of course, is a pressure-driven flow in which you could have either laminar flow or turbulent flow. And so it's unclear if there are any kind of general rules in this sense. There are general rules for microscopic systems. Of course, the pioneering work from Chris Straczynski allows us to relate the work invested to establish non-equilibrium state to its free energy. And this applies arbitrarily far from equilibrium. This is for microscopic systems, of course, in which we're talking about fluctuations. But on a macroscopic system, these fluctuations are less relevant, and we don't have as good of a framework. There are frameworks, of course. Lars Ansager, through his reciprocal relations, was able to relate kind of forces and flows like pairs of these bulk parameters to dSdt to a distance from equilibrium. However, that is also for a system that is near equilibrium. So on the bulk level, we've got relations that are near equilibrium. On the microscopic level, we've got things arbitrarily far. Can we think of maybe how to connect the two? I won't answer that, but I'll add to that conversation. What we want to reevaluate, though, is for many of these parameters that are ill-defined outside of equilibrium, can there actually be information in them, though, that can be related? And what can we do with that information? Can we actually use some of this information to try and determine if there is if there are any variational principles or conservational relationships that we could use far from equilibrium? So what we desire to do is to try and connect microscopic parameters to bulk ones. So on the microscopic level, of course, we understand that microscopic processes at thermodynamic equilibrium are reversible. Out of equilibrium, however, there is a degree of irreversibility. And so what we do in the lab is we disassemble proteins, we reactivate their components, we regenerate interesting and complex dynamics, and then we evaluate the extent to which they are irreversible. One way in which one can do that is by taking a look at correlations in their dynamics, both forwards and reverse in time. So for example, in this movie here, you see the diffusion of particles in a medium. What you probably can't tell is that halfway through the movie is reversed and played again. You can't tell the difference between forward and reverse, then we say it's time symmetric. The probability of any forward path, space, which in this case is real space, is the same as a reverse path. However, if we were to inject energy into the system, of course, to bias it in some way, then the probability of a forward path and a reverse path would be different. And so as you can see clearly in that movie, when the movie is reversed. So using things like information theoretic approaches, we can estimate something like the production of entropy. We can do that for random variables, but we can also do that for spatially extended quantities. So here, for example, we have a reaction diffusion network, one which is not at equilibrium, but not far from it. And again, it's very difficult to see when the movie is reversed. Or we can take a look at very far. What we can do in any event, because of course the temperature is ill-defined, is not get a dissipation per se, but at the very least evaluate the extent to which detailed balance is broken, which is reflected in these forward and reverse paths. And of course we do this to be able to try and outline kind of interesting phenomenological events with these active systems, things like try and predict non-equilibrium changes in material phase or just dynamical phase transitions. So for example, we've done this to show that in that reaction diffusion system, where one can go from, say, a fixed point to a limit cycle, that changes in the production of entropy can actually predict that. What's more than that, with these spatial quantities, we can try and identify actually what are the frequencies that carry that energy. And so this is all what we perform at the microscopic level. But even if we had this information, on a more macroscopic level, on a cellular level, cells will perform functions. They'll move, they'll divide. And so how does the dissipation relate to that? So in '77, Ilya Prigogine suggested that systems could optimize the dissipation of energy for kind of complex assembly. While not necessarily referring to living systems, indeed living systems have an extraordinary variety of complexity. both in the assembly, again, of those protein-based polymers. Here are the same proteins pretty much in different organizations that have different functions in the cell. And of course, they can establish those complex patterns of chemical and reaction dynamics. Furthermore, not only do we not know to what end they influence assembly, but also for what end, for what purpose. Is dissipation in some way optimized, for example, for efficiency, or for a particular purpose like speed or accuracy? So for example, in this work, they look at the motion of molecular motors, which through the consumption of chemical energy are going to move cargo along other protein-based filaments called microtubules. And they change the consumption of ATP, they change the amount of ATP per unit time, and take a look at the efficiency of that process by the force by some velocity, by that chemical potential. And what they show is as they change the amount of ATP, at first the efficiency of that process increases, but then it decreases. So one interpretation is that perhaps the system isn't actually optimized. As you feed it more energy, one might expect it to get better at its performance. But also, we don't really know what our system boundaries are in that sense. Is it a collection of proteins? Is it at the cellular level? Perhaps it's at a tissue level. So really, most of these questions are poorly defined, which explains why we have little information. So then overall, to kind of summarize, again, we know that in equilibrium, we can connect microscopic parameters to macroscopic parameters. For example, by the kinetic theory of gases, we know that particles that bombard a wall will transfer the momentum, and that's going to contribute to a pressure. Equilibrium and equation of state allows us to relate that pressure to other stuff. Out of equilibrium, it's a lot less clear. And so kind of like the overall goals of our lab then are to try and understand how we can try and connect these microscopic processes to macroscopic processes, and then relate that to biological outcomes and see how that can inform on material design. And so in this sense, we actually want to see then for systems that are reversible that should be conserving energy, for cases that are irreversible, what actually happens. So just as a brief introduction to the methods that we use, my lab is organized kind of into different parts. I mentioned that we will assemble biological systems from scratch. We take apart cells, we purify proteins and lipids, and we put them back together in a way designed to resemble the cell and to mimic some of its active behavior. So for example, this is not a cell, but it's an artificial cell with a membrane and that actin polymer polymerized inside. We can activate it through the addition of chemical energy, and we get interesting dynamics. From those dynamics, we can try and understand something like a production of entropy. But in addition to doing things at the molecular level, what we would also like to do is to use bulk or continuum level models to also kind of constrain non-equilibrium systems. And for that, we use interfacial mechanics. And that's what I'll be focusing on today. So what we're interested in are very, very general properties of living systems. We want to understand how things grow. And so we're going to look at the assembly of tissue, a very, very simplified form of tissue, one that's just a bulk aggregate of cells, and also looking at collective motion. And by energetic constraints, really I'm looking to say, can we identify any type of conserved principle or variational principle? So in equilibrium, for things like passive fluids, for many passive fluids anyways, we know the pressure is homogeneous and isotropic. So for example, for a particles in a gas, we know that particles can bombard the wall, transfer their momentum, that creates a pressure. And at equilibrium, we've got an equation of state, PV equals nRT. If that's happening in an extensible container, like a bubble, then we can also relate the pressure to the surface tension of that bubble. In this case, however, we know that the pressure is going to depend on the curvature of the system and on the surface tension. But the surface tension is intensive. It does not depend on the size of the system and is isotropic. So for living cells, I mentioned there were a multitude of active processes. I mentioned that there were ion channels, the assembly and disassembly of the cytoskeleton, and molecular motors. all of those things can cooperate simultaneously to drive the system far from equilibrium. Again, we can establish those gradients and ions. The actin that I've been talking about is this polymer network that lies right beneath the membrane. It's connected together, and one of the things that connect it is those molecular motors called myosin. So these were the three different kind of activities that I talked about. We're going to focus mostly on this one, the one that was sliding those active filaments, generating active stresses. Now, the result of this was that you have pressures inside cells which are non-equilibrated, at least on short time scales. So here what you see is a cell that's migrating through its extracellular matrix. It's different than the one that I showed the last time. And it's doing this through a type of migration called blood-based motion or amoeboid motion. So you see it creating all of these bubbles. What happens is that it will create a protrusion into an adjacent pore. It will expand that protrusion. It will shrink the rear protrusion and thereby move into the new pore. And it does that process iteratively. What makes it non-equilibrated, however, is the fact that this material behaves like a poroelastic material, the actin-myosin membrane composite. Contractility of the actin-myosin complex will drive fluid flow. That fluid flow will couple to the solid, and the deformation of the solid then moves diffusively. And so you're going to have a time scale which is related to the hydraulic permeability of the material. And so that can explain then why you don't equilibrate for things less than that time scale, and why, for example, these blebs don't have to look completely correlated. If it were a balloon, anything that happens has to be felt everywhere. Because it's not non-equilibrated on these time scales, you can have blebs at any time, pretty much in any place. Not quite close. However, we ask for this non-equilibrium system then, well, what then does bound the pressure volume work? If we're expecting this pressure to change and the volume of the cell to change, in equilibrium, if we have an equation of state, we can say directly what that is. But what bounds it if it's out of equilibrium? So to address that, we're going to use a very, very simplified model of an active system, which is a tissue. And I'm defining a tissue as just being an aggregate of cells. So here we see a ball of cells. These are actually cancer cells called sarcoma cells. They have been engineered to be very simple. They are engineered to not produce any extracellular matrix. Cells will do that on their own, which makes things more complicated because it creates the environment that it's in. These cells won't do that. They're also engineered to have strong E-cadherin bonds, and what that is a molecule that binds cells to each other. It makes them sticky to each other. And so all of these cells are very sticky to each other, which can be reflected in the fact that it's round. And so we know that it behaves somewhat like a liquid. It's minimizing its surface area, suggestive of a strong surface tension. It's been shown that if you put two of these things together, they will fuse and become round, and that they can, when put on an adhesive surface, spread very much like a liquid. And so here you see this aggregate binding to a surface. There's a single cell layer thick monolayer of cells coming outwards that kind of resembles a precursor film for a wetting droplet. So this is going to be our model system. So what we want to do is calculate a pressure-volume relationship to see how we can estimate what the pressure-volume work is, to see if there are any conserved quantities. So what we do first is that we calculate a surface tension, and we do that with micropipette. So here we have a little micro needle. This is, I believe, about 100 microns in size. So we apply a pressure which will aspirate the aggregate, pulling it inwards. So it's going to come in some length L. So we're adding this kind of step pressure. So first it comes in quickly, and then it comes in more slowly. And then we release the pressure, and then it goes back. Now, from that, the rate of going in versus going out, we can estimate a pressure. And through that, using Laplace, estimate a surface tension. So what we find when we calculate the surface tension of the aggregate is that it varies from what we expect from a droplet. So our surface tension here is gamma. And what we find is that the surface tension depends upon the size, R0, the radius, of the aggregate. So for small aggregates, the surface tension is large. And for large aggregates, the surface tension decreases. Now, that principal source of activity that I mentioned was coming from those myosin molecular motors, the motors that were pulling on the actin filaments. We can inhibit myosin's ability to use ATP. So we can do that by adding a drug. That drug is called blebistatin. And so what it means is that myosin will bind, but will not undergo its power stroke, which is pulling on these actin filaments. It will not impart these active stresses. So when we do that, the surface tension decreases, but still to a reasonable value, but now it becomes independent of size, like we would expect for a passive liquid droplet. Now we're treating this like a liquid, and the reason we're treating this like a liquid is that while our experimental time scale is 1 to 2 hours, the relaxation time when we switch from an elastic to a more viscous system is only a couple of minutes. And so those couple of minutes are far shorter than the experimental time, rather inversely, our experimental time is much longer than the viscoelastic relaxation time, which means it's a liquid. So in this case, then, we find that the surface tension is non-intensive. It depends on the size of the droplet or on the population, how you would have it. And that the size dependence comes from this particular form of activity. So again, using Laplace, we can estimate the pressure inside of the aggregate from the surface tension that we just measured. What that means is we're going to get a result like we had for the surface tension, but we can turn that into a pressure-volume relationship. So from Laplace, we can calculate the pressure from the images of the aggregate that we had. If we assume it's symmetric around the other axis, we can calculate the volume. So what we find then is that small aggregates have a higher pressure and larger aggregates have a lower pressure. Again, if we were to inhibit the myosin molecular motor activity, the aggregate would inflate. This is a change in the volume. The aggregate will inflate. So if the aggregate inflates, we assume that the same number of cells are there. And so that means that the density must be changing. So then we tried to quantify what the change in density was. So all of these cells are mononucleated. They've got one nucleus. So we spot track the nuclei. And we estimate the density then for small aggregates to large aggregates. Already phenomenologically, you might see that there's a difference in density. If we calculate that explicitly, we find that like the surface tension and the pressure, there is a higher cell density, number of cells per unit volume for small aggregates than for large ones. We have two methods of calculating that depending upon the size. So the 3D means that those are small enough, we can do a single confocal and just count them up. 2D are larger, so what we do is we allow them to spread on the surface like that wetting droplet, and we just count the ones that come to the surface. You have a question?
Yeah. So if you take the cells that aren't treated with blebistatin,
yeah,
and you just kill them with something, like formaldehyde or something like that.
Okay.
I don't know what you. But would the blue curve shift down to the orange curve? Because the orange curve,
blue curve shift down to the orange curve.
So in other words, would that tissue, assuming it still holds together, it should behave just like a normal fluid, which would be like the orange.
Well, if we kill them, then those active processes wouldn't be generated. Yeah, so we wouldn't be generating that pressure. There'd be no surface tension, no compensatory pressure.
So the surface tension doesn't come from the cadherin.
So the surface, I see what you're asking. The surface tension we found comes from the cadherin to a small degree. So if we kill active processes here, the surface tension drops to about 15 to 20% of its value. There are passive contributions.
And those look like a normal fluid.
And those look like a normal fluid.
Okay.
Yeah. So there are indeed passive contributions, but they're less. Yeah. So that's what happens when we kill the blebistatin, is that the aggregates, I think we'll see that a little later, the aggregates remain round, and you can measure a surface tension, but then it is independent of size, like the thing I had in the previous slide. Yeah. So you can probably tell these are different units. This is radius, this is volume, but you could probably see already then that they scale somewhat similarly. This is actually more accurate and is specifically 1.8. just like the pressure, and then 1.6 when we include the whole range. So if the pressure and the density then are scaling similarly, right, in equilibrium, these should really depend on size. I mean, pressure depends on size because of curvature, but not because of the surface tension. These ordinarily would not depend on size, but they seem to have a very similar dependence on size, which means if they plot them against each other, we've got a straight line. And so that a straight line, of course, means it's predictive. And so what we end up getting then is something that looks, oddly enough, quite a bit like our equilibrium equation of state for an ideal gas. So that's kind of bizarre. Of course, that's for a non-interacting system. Here we have a very interacting system. But what it says then, what that really means is that the pressure volume work, or P by rho, is constant. And so that's a work. In this case, that would be something like the work of changing the size of the aggregate. So it should be noted, technically I make aggregates of different sizes. We don't wait for them to grow. We just make ones of different sizes. And so each of these is a completely different aggregate even. You have the same work for changing the size of the aggregate. The work is independent of size, but actually the parameters are not. The surface tension, the pressure, the density all depend on size. Interestingly, however, they depend on size the same way. So in equilibrium, you would get a constant, but it's constant because neither of these depend on size. Out of equilibrium, they're constant, but they're constant because both of them depend equivalently on size. What that also kind of implies then is that we can estimate something like a distance from equilibrium. And that would be something like an effective temperature, which of course the granular field is very familiar with. So could this be a law, a rule for understanding the growth of tissues only when surface tension matters? So of course, again, it's not an equation of state. It wouldn't apply for all systems. Only kind of as important when we consider capillary effects. But if we were to change the myosin again, so if we were to inhibit myosin molecular motor activity, to your question, again, we would get a straight line. So it's also defined by a slope, which would just suggest that that's a change in temperature, an effective temperature. And of course, others have gotten kind of similar values. Yes, please.
Did you try to deplete all ATP in the system?
We didn't try to deplete it because we definitely know that causes problems because of the metabolism.
And you will
All of the stuff I
also
ignore.
the sticking, then probably.
Yeah, so there's a basal level of sticking that should be okay because that's divalent cation dependent. But then actually, if you don't have any active stresses, like at all, blebistatin gets rid of most. If you don't have any, they all fall apart.
Yes. Yeah, that was my next question. If you chelated calcium.
No, well, we have.
Yeah.
But then we
falls
haven't
apart.
calculated that because they fall apart. So this only works for stuff that's round. Times when surface tension matters. So therefore, it's not universally applicable, but yet there's clearly an energetic constraint to growth. So for the conclusion for the first part, we say, interestingly, surface tension, pressure, and all this is size dependent. Ordinarily, these should be size independent, but yet they vary. And the varying is identical. So at the microscopic scale, it's been suggested, for example, that pressure is not a state variable. That it can't uniquely define a state. But yet, if there's information in it, for example, like this anomaly, like the size dependence, that information could be correlated to other ill-defined variables and still define. I call it, it's not an equation of state. In other work, we've looked at collective migration of cells, compared it to the wetting of a liquid film. And that way you kind of balance a gain and adhesion over time to a dissipation rate, and you get a scaling relationship that's pretty much identical to what you would expect for a passive liquid, except cells are choosing between two different active mechanisms. And they actually choose just the right balance of those active mechanisms to make it look that way. And so in that case, we find not that there's a constant work, but for this dynamic process, we find that there's a constant power, a constant work per unit time. Okay, so next we want to talk about not just more static process like the size of aggregates, or I guess dynamic if we call it growth, but we can look at collective motion. But I'm going to liken it to a phenomenon that I'm sure you've seen, which gives away most of the interesting elements later, which are Marangoni flows. So for probably most of our favorite beverages, evening beverages, hopefully, we know that surface tension will draw liquid up the wall. It'll evaporate. The surface tension will increase. It'll accumulate, get larger, fall back down again. Actually, for coffee ring effect, I believe that's a morning beverage, right? So perhaps for both of them, it actually matters. You could also have movement in a droplet. So here in a droplet, you could create a gradient in surface tension. The gradient surface tension could be evaporation or an additive or something like a detergent. I always think of the commercial that was pretty popular when I was growing up of like Palmolive. They would drop a detergent onto the kitchen sink that was full of water, and everything on the surface would move apart very quickly. So you can have flows that are driven by surface tension gradients. So I choose to look at surface tension gradients because I'm trying to convince you that surface tension matters. So if surface tension matters so much, what happens if we make a gradient? I gave it away already. So here is a cell aggregate. What we're doing here is we're using a laser to kill a single cell or close to it right on the surface. This is an image of the F acted inside. There's some alignment. You see that the cells are kind of elongated on the surface, a little bit less so inside. And so here's a movie that plays a little faster. This one is a little slower and has PIV vectors on top. So what you might be able to tell is that when we ablate and kill a single cell, I'll start from over here, the side peels back, the center moves forward, then the sides move forward and the center moves back. And so you have this kind of toroidal pattern of motion, which is kind of a hallmark of Marangoni flows. So there's a few things to note with this process. I'll let it play again. Again, things peel back, move forward, move back. So if you note the time scale, however, it's actually extremely fast. For motions that we're used to in cell biology, we tend to think about cell migration, which typically occurs on the time scale of minutes to hours. Here you've got very fast and extraordinarily highly correlated motions. So for example, they would look like this if we separated those two time scales. So again, we have this aggregate, we kill a cell here, they were all tensed by that myosin activity. And so I kill one, we pop it open. And again, we get this toroidal thing here where you have the top moving back, center moving forward. Just some seconds later, it completely reverses. So here you can take a look at the velocity. It retracts really quickly. It then goes back the other way and then has kind of a slower return. But even that is all done by 100 seconds. If you were to add up these displacements, it actually does go back to basically where it started. So the behavior is completely reversible, and it reverses in just about 2 minutes. Again, if we were to get rid of blebbistatin, get rid of myosin activity with blebbistatin. So again, we've gotten rid of the majority of the active stresses, the non-equilibrium driving. It's still around from the passive contributions, but there are no flows. You have no correlated motion. In fact, all you have is just kind of the motions of a very unhappy tissue. So we see no indications of toroidal motions. Now, before, we were saying that the aggregate behaved as a fluid. We compared the timescale of the experiment to the viscoelastic timescale. It was much longer, so we called it a fluid. In this case, we were showing that you have motions that are just a few seconds long. So the viscoelastic relaxation was a few minutes. But now, if we compare each of these time scales, so the initial relaxation, the reversal, even the kind of slow return to the original configuration, on average, all of those time scales are shorter than the viscoelastic time scale. So that would actually suggest that for those motions, the aggregate is actually elastic. But of course, we only understand Marangoni flows for fluids. But it seems to be happening in an elastic solid. So we could actually test that a little bit further because we could just plain try using different constitutive models. So we can, through the PIV, we can calculate a strain and a strain rate. I didn't show it, but from those calculations of our surface tension, we actually also calculated the elasticity and the viscosity. So we could actually just take the elasticity by a strain and the strain rate by a viscosity and calculate our surface tension gradient. We can integrate across it to calculate our delta gamma. And from that, we can get two things. So we have our viscous constitutive model. So this is our measured viscosity with our strain rate versus our elastic constitutive model with our measured elasticity and our strain, our maximum strain. And what we get are two sets of values, magnitudes, as well as the fact that if we were to integrate around, it should come close to 0. And so if we compare those, our viscous and our elastic models, for both the forward and the reverse, we see that they're better fit to the elastic models in both cases. So it really does seem like the aggregate is behaving like an elastic solid, even though you're getting this behavior.
The different curves are different clusters.
The different curves are different times, I think, during the motion. So we integrate over different periods of time. So, I guess then we're talking about the surface tension of a solid. Before we were talking about size-dependent effects. So what we did then was take a look at how different parameters depended on size, like the rate at which these things moved, those different time scales, but then also our delta gamma, or actually, our maximum delta gamma, because that changed in time. And so if we compare the delta gamma to size, we see that small aggregates have a larger delta gamma. Kind of as we had said with our micropipette measurement, where we were calling it a fluid, and we calculated a liquid surface tension. Now we're technically calculating the change in surface tension, but for a solid, and we find that that indeed changes with size. But if it were actually a solid, then it should scale with the strain. So outside of ablating it, we can calculate kind of like the steady state strain of the system, what's kind of programmed in there beforehand, by looking at the shape of the nuclei. So I mentioned that on the outer side, the nuclei seemed more elongated than the ones in the middle. So we could turn our aspect ratio into a strain, and then plot the strain of the outermost layer versus the size of the aggregate. And so the nuclear strain, again, varies with size. Small aggregates have a higher strain. The cells are more elongated than large ones. And again, if we inhibit myosin activity, the strain goes down. It's not 0 because of the passive effects, but it is independent of size. And so again, if we were to then get rid of size, pretend that we've never looked, and plot the strain versus the stress, we get a line. And the slope of that line, because we're talking about surface properties, should be something like a surface modulus. Interestingly, the magnitude of that surface modulus is comparable to other stuff. For example, like PDMS, other soft gel-like things. And so we have a constant surface modulus, but what is a modulus? It's an energy density. So again, now we have size-dependent properties, but can be characterized just the same way as we would for a passive system. We would have measured a surface modulus had we never looked for size. I mean, a constant surface modulus had we not looked for size. One reason I think this is interesting is because the traditional way in which we think about migration is again from the single cell perspective, where cells will generate surface stresses at their surface. They'll reach and grab and pull. They'll drive their own migration. They can do this together. And so collective motion is always thought of as, let's add stresses between them such that they coordinate, but still walk on their own. However, there is an opportunity to consider a broader perspective. I mentioned that this was really a fast motion. If we look at typical migration rates for single cells or even sheets of cells, they tend to be lower than a lot of the collective motions that we see during development, which in some cases already look like wetting processes, things that might be driven by surface effects, surface tensions. We can even see remnants of that at various stages in a more complex development, like with Drosophila. So could it be possible then that we can drive motions, even in complex scenarios, by altering surface tension gradients? It has been shown that the tissue surface tension and as an equilibrium property can determine the sorting of cells. That was done by Malcolm Steinberg at Princeton in the 70s. And here is basically a dynamic version of that. So here you can see fast flows on the right, slower flows elsewhere. So if we can control locally these interfacial stresses, we should be able to generate migration-free motion. So overall, showed that gradients in solid surface tensions can drive Marangoni-like motions. I have to call them motions now since they're not closed. But again, the stresses and strains are size dependent, and that's going to yield a constant surface modulus. So there's a lot of things. I mentioned wanting to connect microscopic to macroscopic behaviors. I only showed you the macroscopic. But in general, we know a bunch of funny aspects. So we just said that at the microscopic scale, processes are time reversible. Are time irreversible. At the macroscopic scale, I showed you reversible processes, right, due to activity. Again, an actual equation of state for a thermodynamic system is for a system that's not interacting. But it's not an equation of state is for highly interacting systems. Also driven by an effective temperature, but that kind of implies we had something like Gaussian dynamics at a smaller scale, which we don't have. So there's a lot of interesting problems to continue with discussions. And so in summary, I began by trying to suggest that there were ways that we could try and correlate poorly defined variables out of equilibrium for the sake of trying to identify conserved quantities. This has been done, but the assumptions are that they have to be near equilibrium. We show that you can be away from equilibrium and you can violate equilibrium constraints and have anomalous properties, but that those anomalies need to themselves be correlated. Also, over and over again, those anomalies, if we didn't look for them, we wouldn't find them. You would measure passive-like properties in each of these cases if we never looked for a size dependence. So I always get asked in the end, is this general? Probably not. It's a measurement for the most part, an experimental measurement. So it means it's valid for this case and this case only. So I say very generally that it means that it's not impossible. And so with that, I would like to thank our group. Actually, many of these people are gone by now, but this work is largely a result of some very talented postdocs, especially Vikram Yadav and Suleiman Mohammed. A lot of our stochastic work is through, he's graduated now, a very talented student, Danny Sierra. I've had some really great interactions with theorists and other people. Very notably, my good friend Eric Dufresne. I'd like to thank the funding and then you for your attention.
Thank you very much. Aditya, sorry. She was.
So when you ablated the cell in the active cluster, was there a reason it translated to the opposite side?
When I ablated the cells on the cell aggregates,
Yeah, they move in the other direction. Whereas if they're in some medium and if it's small enough, I would have assumed them to move in the direction of the wave.
Oh, you mean the whole thing? Actually, I'm not really sure what happened to the movement of the whole thing. It moved backwards.
Yeah.
Is it scalloping, perhaps? I'm not sure. Yeah, I don't know.
So when, yeah, actually, I was wondering that too. When the cell died, did it pop and like blow a whole lot of stuff into the
It does, yeah.
Yeah, and so could it be like basically like a jet plane?
Yeah, I guess so. So it moved backwards, I guess, right?
Yeah.
So yeah, I guess that's possible. Yeah.
Paul, you had a question?
But then did it go back once it? Didn't look like it did.
What if you measure surface tension just with contact angle?
We haven't done that
I would
because
think you just measure a property of the actual surface rather than something different from surface tension, which is sort of a macroscopic thing that you're getting from your aggregate.
so we would need to get what the gamma for the solid liquid would be, like an adhesion energy, which I guess we don't have.
Well,
To get that surface tension, we
so
need the
you
other
don't know that,
components.
but it should change with size if you're right.
Yeah.
And therefore, you really have a contrast. The surface
Well, I think the main
properties-
problem is that it's going to we would have to find a condition in which we didn't have complete wetting.
Well, another liquid, you think it's going to wet, completely wet another liquid?
So for looking at the contact angle, we would do that on a solid surface that's coated with adhesive
You
protein.
could also just put it at the interface between two liquids.
Oh, I see. Yeah, I guess we could. Yeah,
Or mush it against the cluster of a different kind of cell.
yeah. We have been starting to do that, but for other purposes, since they have different surface tensions, we're actually putting different ones of different sizes together.
You make it sound evil.
Or the way how you hold with your pipettes, right? You could hold two and putting them against each other.
Yeah.
But it's, yeah,
If we had that
already the first one
capability.
is, already the first one is very, very tricky, yeah.
Yeah.
Yes, Irash.
I have a very basic question. So you told us that when you measure these slopes of pressure versus density, you can get this estimate of an effective temperature.
Yeah.
What kinds of temperatures are you measuring? Like how far from room temperature are these?
Orders and orders of magnitude. Many.
And when you do blebistatin and you measure that slope, do you get room temperature?
No, so you're still going to have a lot of other active processes. So it lowers it by like 2 orders of magnitude. So there's still orders that are not accounted for there. So it doesn't go down that far actually. Only say like 100-fold. There's many
and
more
you're
to
still
go.
far away.
Many more to go. Yeah.
Wow.
I think that probably has to do more with that idea of like the 1%. Perhaps.
Oh, right.
Is that there's probably still some factor for what overall the cell is doing that may not be mechanical in nature per se. I mean, we are looking at a pressure volume, I guess. I guess it has to be mechanical in some way. But yeah, there's more to go for sure.
Any more questions? Yes, Glenn.
Yeah, so I actually now have 2 questions. So one is just came up, which is you said that the two blobs will merge together. So is that another way of measuring surface tension? When
It
you have
is.
the neck, but that could be a different surface tension maybe because it's back to the liquid one, or
It's very liquid, so to do that takes like 40 hours. So that would definitely be probing a different regime for sure.
yeah, this is kind of what my original question was, which is that when you were saying this, the ablation experiment is elastic, and the pipette it's a liquid property, but I was wondering, is it because you're shearing it or because it's a slower process and so it's just elastic on minute time scale? And like when it wets, is that because it's just a long process or is it also doing something different?
It's probably also doing something different because the process of adhesion itself activates all kinds of things on the cell. So we use the time scale argument because we're not doing any adhesion. You're probably asking the pipette is coated so that there's no adhesion. So it should slip with regards to the wall.
But then it happens faster, or you can see it on a shorter time scale, but you're also applying a shear force, so that it change. You have shear.
You have shear, you have extension, you have compression. With micropipette, you have all of the above. So we don't get too specific for that reason, actually.
Have you looked at the time evolution of the leading edge of your wetting on the surface?
Yes.
well, from that you can get actual surface tension as well, right?
If you know, again, if you know your droplet surface
Yes.
gamma,
Yes.
which we
I
don't.
see. Yes.
I think somebody, I mean, probably
How big
Francois
are these
says,
aggregates?
like
Right.
between 50 and 500 microns,
Mhm.
couple hundred microns on average.
So that's even tricky actually, even to get the imaging, so you have to do like interference or something to get the angle
Oh,
out.
you can image it, but then to get that dynamically, you would need to, for example, already have at the very least, so you need the gamma, say, or the viscosity.
Yeah.
So you'd have to measure one of the two. So you'd have to either measure the adhesion, like the pulling
Yes,
off
yes,
force,
yes.
or you'd have to somehow measure the viscosity, which itself is
Yes. I was thinking for the
complex.
contact angle, right, you would want to have actually the basically XC plane, right, imaged. So this might be tricky for such a large aggregate.
So you can measure the gamma without the contact angle for complete wetting, because you can just look at the dynamics of it and balance the gain and adhesion with the viscous dissipation. If you did it young Dupree, you would need to have something that isn't fully wetting, and they're always fully wetting. It never goes and stops.
That's that's very cool. Any more questions? If not, let's thank Mike again. And is it true that we have a reception as we used to have in old times? So please, everyone, come to 871. We're returning to normalcy. That's fantastic.